Welcome back to MLE Masters, your destination for mastering Step 1, one high-yield concept at a time. In this video, let's dive deep into molecular techniques, a crucial part of your USMLE preparation, often tested through clinical vignettes, buzzwords, and applied diagnostic reasoning. Let's begin with a quick introduction. Molecular techniques are powerful diagnostic tools used in modern medicine to detect genetic, infectious, and neoplastic conditions. They are often seen in clinical labs and Step 1 questions, especially when tied to disease pathology. Understanding their mechanisms and clinical applications will help you answer test questions with confidence. First up is PCR, polymerase chain reaction. This technique is used to amplify small amounts of DNA into millions of copies. The process involves 3 key steps: denaturation, heating the DNA to separate strands; annealing, cooling to allow primers to bind; elongation, DNA polymerase synthesizes new strands. Clinically, PCR is used to detect pathogens such as HPV, TB, or genetic mutations like BRCA1/2 in cancers. It's a core lab technique in both research and clinical settings. Next is RT-PCR, reverse transcription PCR. This is a modification of PCR used to detect and quantify RNA expression. The RNA is first reverse transcribed into complementary DNA, which is then amplified. High-yield clinical use includes COVID-19 diagnosis, HIV viral load monitoring, and oncogene expression studies, commonly asked on Step 1. Then we have the famous Southern, Northern, and Western blot techniques, remembered by the mnemonic SNOW drop. Southern for DNA, Northern for RNA, Western for protein. These tests are commonly used in diagnosing genetic disorders, confirming HIV, and identifying specific protein markers in cancer research. Let's continue with another high-yield tool, ELISA, or enzyme-linked immunosorbent assay. ELISA is used to detect antigens or antibodies in serum samples. There are 2 main types: direct ELISA, where antigen is detected, and indirect ELISA, where antibody detection is key. You'll often see ELISA in HIV screening, hepatitis testing, and in autoimmune disease panels, like anti-dsDNA in lupus. Next, we have FISH, fluorescence in situ hybridization. This technique uses fluorescent DNA probes that bind to specific regions on chromosomes. It is particularly useful in identifying chromosomal microdeletions, like 22q11.2 deletion in DiGeorge syndrome or Philadelphia chromosome in CML. If you see a vignette describing abnormal karyotype or fluorescent tagging, FISH should be top of mind. Now let's talk about microarray analysis. Microarrays allow us to examine thousands of genes at once to assess expression levels, detect SNPs, and identify copy number variations, CNVs. Clinical applications include cancer profiling, developmental delay diagnosis, and pharmacogenomic testing. Then comes the game changer, CRISPR-Cas9 gene editing. This revolutionary technology uses a guide RNA and the Cas9 enzyme to introduce specific cuts in the DNA at target sites. It's now being applied in gene therapy trials for sickle cell disease, beta thalassemia, and other inherited disorders. It also raises ethical and future medicine questions, a hot Step 1 angle. Up next is flow cytometry. Flow cytometry analyzes physical and antigenic characteristics of cells using fluorescent markers and lasers. It's the gold standard for leukemia and lymphoma typing, especially identifying CD markers like CD19, CD20, CD3, etc. It's also used in HLA typing, monitoring immunodeficiency, and cell population analysis. Let's wrap with a summary table. As always, the key to scoring high on Step 1 is clinical integration. Don't just memorize facts, understand how they apply. Here's how you'll see these techniques used on test day. PCR, detect infectious diseases like TB, chlamydia, HPV. RT-PCR, assess viral load in HIV or detect oncogenes. ELISA, screen for antibodies in HIV, hepatitis, autoimmune workup. FISH, identify chromosomal abnormalities in syndromes or cancer. Microarray, diagnose unexplained developmental delays, analyze gene expression. CRISPR, understand its role in therapy, ethics, and research. Flow cytometry, identify immunodeficiencies, leukemias, lymphomas. Keep connecting molecular tools to real clinical scenarios. That's where the magic of high yield lies. If you found this helpful, don't forget to like, share, and subscribe to MLE Masters, your daily dose of Step 1 power-packed reviews. See you in the next video.